Good afternoon, ladies and gentlemen, our distinguished guests, faculty members, students, and our online audience. We are very pleased to welcome you all to this very special occasion, the Obaid Siddiqi Memorial Oration. Today we gather not only to commemorate the life and work of a pioneering scientist, but also to reflect on the profound impact that Dr. Obaid Siddiqi has left on the scientific community, particularly in the field of genetics, neuroscience, and beyond. Dr. Siddiqi started his career as an agricultural scientist at Aligarh Muslim University. During his PhD at the University of Glasgow, his work led to one of the first reports of its kind which mapped the fine structure of a gene in the fungus Aspergillus. His postdoctoral work in Professor Grent's lab at Cold Spring Harbor led to the discovery of suppressors of nonsense mutations in E. coli, which was in turn instrumental in discovery of nonsense codons. Upon his return to India, he set up the molecular biology unit at the Tata Institute of Fundamental Research at Bombay. Along with establishing the basic principles of bacterial conjugation and lateral gene transfer, his lab made seminal contributions in the field of Drosophila genetics, providing an understanding of the nerve conduction, synaptic transmission, and also in the field of chemosensory neurogenetics. In recognition of his work, his scientific contributions, he was conferred country's prestigious civilian awards, the Padma Bhushan and the Padma Vibhushan. In addition to his research, Dr. Siddiqi immensely contributed to the scientific community by training a large number of students and building several institutions, either in advisory role or in administrative roles, such as the biology unit at the IFR and NCBS as the founding director. He has also helped in framing the initial research direction for our institute, NIBMG. Quoting Dr. K. Vijayaraghavan, there are the daring few who define new intellectual quests and whose courage and leadership create a culture, the nurturing of which makes us all feel special. With this, we'd like to inaugurate today's event with an invocation by Mr. Supratim Ghosh and Ms. Riya Deb.
Hello.
Hello. Hello. Sound
Hello.
এটা না বাড়ি দাও তো তুমি তো সাইন আছে একটু দূরে দূরে সরে যাও না
सर्वेषां स्वस्तिर्भवतु
सर्वेषां स्वस्तिर्भवतु
सर्वेषां शान्तिर्भवतु
सर्वेषां शान्तिर्भवतु
सर्वेषां पूर्णं भवतु
सर्वेषां पूर्णं भवतु
सर्वेषां मङ्गलं भवतु
सर्वेषां मङ्गलं भवतु
ॐ सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् ॐ शान्ति शान्ति शान्तिः
Thank you for the beautiful rendition. I would now request our director, Dr. Sagar Sen Gupta, to kindly deliver the welcome address and introduce our speaker.
So, good afternoon, friends, colleagues, students, everybody who is here online or have joined in person. It is indeed a great pleasure for me to have this year's 12th Obaid Siddiqi lecture, memorial oration. And as Alpana had rightly indicated, the contribution of Professor Siddiqi to the fields which he touched are immense. Genomics and genetics actually go hand in hand, and in that way, it is entirely apt that NIBMG is actually organizing the Obaid Siddiqui Memorial Oration, and this has been going on for a number of years, and today it is 12th iteration. It is also apt today to have the speaker with us, Dr. Shantanu Chowdhury from IGIB New Delhi. Shantanu is no stranger to NIBMG, having been associated with NIBMG in many ways, in many forums, in many capacities. He has given direction to NIBMG's research programs over the number of years. He has established strong collaborations with multiple NIBMG faculty members. But even then, as a formal introduction as a director of NIBMG, I have to introduce my friend and collaborator, Shantanu. And it is really a pleasure that he has agreed in a comparatively short notice to come over and give the Obaid Siddiqui talk. So after his PhD in IICT in Hyderabad, Shantanu did his postdoctoral research in University of Nebraska and at Max Planck Institute of Terrestrial Microbiology. He came back to India and joined IGIB in 2002. His group is one of the pioneering groups which studies the role of, if in a broader way, studies the role of transcription and chromatin architecture, especially with certain amount of emphasis on the telomeres. He studies the role if I have to go in more granular details, he studies the role of DNA secondary structure, which is called G quadruplex, and how the G quadruplex actually affects the transcription. G quadruplexes are normally present in the telomere ends, but his group was the pioneer to show that there have been G quadruplexes which are present in the non-telomeric regions of the genome also. And his lab was pioneerly involved in trying to understand the functional vis-a-vis the G quadruplex in the telomeres and in the interstitial, in the quadruplexes which are not present in the telomeres, and how they affect the gene regulation. And these studies actually led to a development of a new class of small molecules which actually are able to regulate the functioning of these G quadruplexes. And he has extended that work to how these G quadruplexes can not only extend the functioning of the gene regulation, but also a much more fundamental process of cancer metastasis. Shantanu has received numerous awards and prestigious fellowships. He is the recipient of Swarna Jayanti Fellowship. He is the recipient of National Bioscience Fellowship, National Bioscience Award for Career Development, Excellence in Drug Research Award, Life Science category, and also, of course, twice he is the recipient of the Wellcome Trust DBT Fellowship. He received the Shantiswarup Bhatnagar Award in 2012 for his outstanding contribution on the mechanisms of gene regulation by DNA secondary structures in diverse cellular contexts. I cannot find a person who can be more suitable to give the Obaid Siddiqui Oration this year. And it is a welcome that I would extend to Shantanu to come over and talk about his work and in the context of his lab work and also other programs which are going on in his lab. Thank you, Shantanu.
Thank you, Shagur, for a very, very generous introduction, if I may say that. In both ways, I have known Shagur for at least 2 decades, if not more than that. Probably more than that. And so is true for NIBMG also, as a community, as faculties, as friends. I know many of you here, and it gives me immense pleasure to, a little overwhelming in some ways, to stand here and stand here as somebody who is giving Professor Obaid Siddiqi's memorial talk. And I completely understand the gravity and the context of this talk, and I would like to really make justice to this platform, to this institution, and in a larger sense, to the community of scientists who have followed Professor Siddiqi around the times that he started. And if one takes a trajectory from there and and looks at the continuum of what has happened from there on, I think it's a colossal adventure which is ongoing. Every day we face this adventure. We look at different in this we look at different facets of this adventure, encounter challenges, and I think we make the most of it. Therefore, we also come to situations like this where we are we're talking about our work, we are appreciating each other, and we play a part in a very large, in some ways, cosmos of things. So coming to today's lecture, I would like to do two things that Shagur briefly alluded to. One is, talk of this collaborative exercise, which is what we've named is as the Breast Cancer Genome Atlas of India. And it's an endeavor in bringing hospitals, labs, analysts, as you will see. I have the privilege of speaking here, but there are about 120 different people, starting from clinicians to students, to lab assistants, technicians, computer scientists, analysts, logistics people who are involved in this work. And it's in some ways a big thank you to everybody who's involved, and which gives me this opportunity to stand here today in front of you. And also, what is the outcome from this, and what else can be done. But what I'm also going to do in the later half of the talk is talk about something which is more inbred, more inside-looking, more granular in the kind of science that happens in my lab and has happened over the years. And some of you, I'm sure, are aware of that. But I will tell as narrative stories, and hopefully engage you in those stories, and we will take in the interest of time, we will take questions at the end of the talk. That way we can have it in a much more methodical way. But please do note down your questions. I'll be happy to engage with you on any question that you have, because all you'll see is snapshots of, in the time that we have, we'll talk about snapshots of the work and just giving you an essence of what is going on and what is that is coming through. And I must not forget to mention that I also am fully aware of, talking about cancer genomics in some way, the mecca of genomics in India is this institute. The oral cancer genomics and stuff that happened here has in many ways encouraged what I'm going to talk about today. And that deep experience, it still contributes to the work, which I will definitely point out as I go ahead through many of you who are actively contributing to this work. So therefore, it's in some ways a very interesting way of saying thank you to the organization, to the institute called that you are all here for. Now, we call it an atlas because it is basically making a landscape of Indian breast cancers. And obviously, when you make an atlas, there are some takeaways. One of those takeaways I will cite as an example of many other things that are ongoing, but I'm going to talk about one of them. And a little bit of pretext is important, but let me come to that in a minute. And as I said, the second part of the talk will be on this telomeres and how this impact genome-wide gene regulation. And the specific case study that we'll be talking about is of how telomeres can auto-regulate their synthesis, and which it's turning out to be a very fascinating area. And I'll hopefully be able to share that fascination and interest, talk about it in an engaging way. Let me give a little pretext to the talk. The first talk about the Indian breast cancer genome atlas. What is the need is a question that I'm asked often. What is the need for spending so much, because it's a high-cost program? So the way I try to explain it to myself is what I'm going to share with you. That as you are aware, many of the genetic tests for cancer prognosis, a clinician today, more times than not, uses a genetic test to decide how to treat a patient. It is true for many diseases, particularly cancer. And if a test could tell us, the doctor, to go slow or go heavy, or do a very strong surgery, or do a little moderate surgery, or a radiation is required or not required, because these are not only costly, cost is one part of this, it takes a huge toll on the human life, and therefore, the quality of life, the quality of family, the quality of the society in some sense is compromised by a wrong decision or a misdiagnosis. So while the rest of the world is making leaps and bounds in trying to making this genetic tests and using these genetic tests, we are importing these tests for Indian patients also. Sometimes doctors tell me that we have to prescribe because there is no Indian test. And now all of you will understand, if a genetic test is based on a population which is not Indian, it's not difficult to imagine that the efficacy of that test might be compromised. So exactly that happens, and this is true for some I'm talking about breast cancer. So breast cancer prognosis tests cost somewhere between 3 lakh plus per test, and doctors prescribe. And while the certified, all the reported efficacy of these tests are about 90%, which is very good. 9 out of 10 patients are treated correctly. But that is true for the Caucasian population, UK, US. When this test comes to India, doctors say that 4 to 5 patients sometimes, and sometimes not even that is what is my personal experience from the 100 tests that I have prescribed in the last 6 months or so. So it's not difficult to understand why that happens. But in the absence of another test, which is based on your own population, you can't tell the patient that I'm not going to give you that test, because that's the last straw that the patient has. And sometimes the patient Googles and comes and says everywhere they are giving this, why are you not prescribing it? So this is one of there are many motivating factors. This is one of the motivating factors, which is based on sheer molecular understanding of how intrinsic a genetic test is to the population in question. So therefore, that was the genesis of this program, that if you don't understand the molecular basis or the molecular underpinnings of the disease, how do you develop tests? Hopefully this exercise will lead to something like that. With that pretext, what we decided is we will take breast cancer patients from across India, and this was when this is how we proposed the work, and we'll try to put all that data together. And then there were people who were naysayers who said that, what is that extra that you're going to find, which is not already there in 7,000, 10,000 breast cancer patients that are already there on TCGA. I did not have a great answer for that question, except saying that you never know unless you do. So now I have much more intricate answers for that question. But that was the day when we started about a little less than 3 years back. So here is what I told about, this is all about partnerships, this is all about hospitals, this is all about different kinds of people coming together and making sure that all of them have a single point, pointed question in mind. So we started with 2 hospitals actually. This is Saroj Gupta, which is the Thakurpukur Hospital in Kolkata, and Tata Memorial in Mumbai, and also third hospital joined, which is Rajiv Gandhi. Rest came a little later, Rajiv Gandhi Cancer Institute and Research. This is another hospital in Delhi. And as you can see, many others joined, AIIMS, Sri Shankara, and these are the institutes, IGIB is where I come from, CCMB, this is IMTech, this is IICB in Kolkata, and of course, Brick and IBMG, and I'll come to more details of how an IBMG is contributing to this. And we started with some very simple discussions actually, but we involved a very well-known medical oncologist. Dr. Siddiqi Gupta is also not new to this institution. He has been a part of the SAC of this institute, if I'm not wrong, for several years. So Tata Memorial. Then Shamir Bhattacharya is a chief surgeon in Saroj Gupta. Dr. K.S. Gopinathan is chair of the monitoring committee of the institute of this program. He's from a Bangalore hospital in Bangalore, and also Karthiki is here. Karthiki is also a part of the monitoring committee, and they have really supported handheld, and the monitoring committee has been has been a big support and guidance in trying to sort of make sure that this colossal exercise keeps moving and moves hopefully in the right direction. And then others joined, SBS Deo from AIIMS, who's the head of surgical oncology in AIIMS till about 2 months back. Now he's in Apollo Hospitals. Anurag Mehta, Juhi Tehal, V.S. Srinath from Shankara Hospitals. So some names I've taken. And again, nothing could have been possible without, this is my core sequencing team. As you can see, all women power. Behind the scenes work, Dr. Divya Khanna, special mention because of the effort day in and day out that is put into just managing the program, hospital samples, then collection, making sure that the logistics person has come on time, etc., and also all the sequencing, library prep, everything happens at a single center. Sequencing is in a couple of centers. And a big shout out to this team, without which again, not have made any progress. And without Nidhan's support, Shubhrato, Arnav, Hriday, they've been really, right from the beginning, have been, yes, we are in. And we put all the understandings happened later, the paperwork happened later, the work started earlier. And that's a lot of trust, I must tell you. That is based on a lot of trust. And I'm ever grateful for for the contribution that this team continues to make as we speak. And also on the lighter side of things, the harder questions can be directed towards Nidhan, who is right here, Nidhan Subroto Arnav. And Hriday is now in IGIB in Delhi now. But they continue to contribute very, very strongly to this effort. So what is the status? The snapshot of what is the status about operation started in about November of 2020. 2. So that's about 2 years and a few months, maybe. And what we've been able to do is we wanted to collect tissue, but with adjacent normal. So most of the tissues, more than 800 of this is, about 50 are without adjacent normal, where we have blood as normal. But mostly it is with adjacent normal, plasma and serum. Currently, there's about 720 samples which are processed. We have 600 plus which have gone through whole genome sequencing, and transcriptome is lagging a little behind, but we'll catch up in the next month. And this happens a little faster than the WGS. So hopefully that will be 600 in a few weeks now. From now, so that's the situation we are at now. And obviously the analysis and the washing of the data and making the QC, etc., some fall off. So the analysis numbers are a little lesser than this. But nevertheless, we have analysis from quite a few, more than 350, almost 350 patients for which we have analysis. The first snapshot that I'm going to share to you is the mutational landscape, because how does this, and also answer at the same time, how similar or how different are we from what is the global scenario. So at present, all this happens, the mutation happens right in NIPMG under Nidhan's direction. So this is TNBC is triple negative. All receptors are absent. The 3 primary receptors that are found are absent. These are HER2 minus out of the 3 different receptors that are used to categorize breast tumors, estrogen, progesterone, and HER2. So this is HER2 minus, and this is HER2 plus, and ER/PR positives. And these are about 55 for which we have still not received for some reason or other, we have not received the characterization, or we were not happy with the pathology and we sent it back for the pathology. So this is why it's written as NA. And as you can see, if you are familiar with the landscape studies that have been published for any cancer across, you would see some of these players as major players in many cancers: TP53, PIK3CA, MYC, ERBB2. These are, in the halls of tumor mutational landscapes, these are very strong pillars. You will see these pillars always there. Doesn't matter what tumor you are talking about. So they happen to be right there. So you see most of these mutations are there, and you can see that they are also, irrespective of what type of tumor you are seeing, these mutations are pretty common. But what comes out to be interesting is this mosaicism that you see. That's a lot of patients have. Especially here, you have less than 50%, about 30% patients are TP53 positive, but then there are many others who are TP53 negative and have something else going on. So this is true here also some. So, you see this landscapes more or less almost everywhere. There are a few things which caught our attention, and we try to focus on that a little bit here. So this is Indian Breast Cancer Genome Atlas. This is the study that we are doing, and this is what this is the TCGA data. And what Subroto, Arnab, and Nidhan were able to show is that, if you take TP53 and if you take PIK3CA and you look at the percentages, TP53 not so much, and this TCGA also is, I think these are all triple negative. We are talking about triple negative in both cases, both Indian and the TCGA. And difference is captured here. If you go to PIK3CA, you see that the mutations percentages is about 35% here, but in TCGA it has gone down. So there are differences. We are not finding the same. We are finding probably a much more spread out things. Too early to say. The numbers are also very low, but we see small shoots of differences that are coming up, and hopefully these shoots will be strong trees, or or not. Either way, it's information, and we'll need ways to understand where these differences are coming from. And another thing that is not sort of here, but we can talk about it later or in detail, but something to keep in your heads. We also don't, though we have put the mutations here, we sometimes realize that they're not the same mutations. Genes are same, but we are probably seeing different kinds of mutations. And that's in the context of biology, that's a very, very interesting tenet, or very interesting foundational information, which can have many, many different ways of looking at what is the underlying biology and how is that compromised. Now, as I said, I'll give you a very small peek into what we are trying to do with this data. Apart from having this resource out there for hopefully millions of people to be able to use, download, use it for, and hopefully some of you can put instead of TCGA, write IBCGA in the days to come, that this data is also analyzed from IBCGA gene, RNA expression analysis has been done, or the mutations have been taken from. Right now, we write TCGA. Hopefully, we'll be able to write about the Indian breast cancer genomes also beside that. But beyond that, how does it contribute into something that a patient can use or a clinician can use tomorrow, which is again a very important and very critical part of an exercise like this. So we initially started with triple negative breast cancer patients, and these were also tissues which were fresh frozen but stored for the last 5 years. And many of these tissues, when we went back and took it out from the repository and wanted to do sequencing, we could. The RNA and DNA were of interesting enough, good enough, adequate quality to allow us to do experiment, because you need at least 5 years of follow-up. Did the patients have recurrence? And about 5 years means the tissue is collected 5 years back. So therefore, the tissue has to be stored carefully. And also the normal tissue has to be there for you to be able to say anything about this. So in this case, we had about 90 triple negative breast cancer patients with 5-year follow-up. 14 of them had recurrence, and 76 had no recurrence up to 5 years. And recurrence was within 3 years. And non-recurrence is more than 5 years of these patients have had no recurrence for almost 5 years. Again, small numbers, but the idea was, can we use this as a discovery cohort? So the first thing and when we talked about this with our clinicians, there's something very interesting came out. I was looking for genes which would contribute to recurrence, but they flipped the question over on its head and said, no, let's look at the gene signature which is associated with disease-free survival. Let's look at this 76 more closely. Let's put the question on its head. We would like to know more of what is helping a patient not recur. I think, and that made a lot of sense. And this is where clinicians, sort of give you great, into the same question now talked about in a different context. Sherry is my colleague in IGIB, and her team is doing this whole transcriptome analysis is done in her team. And obviously, they cross-talk a lot with Nidhan's team here. And so the rest of the data is about doing this. This is just to put everything together and right there to sort of show you that all this context can be used as discovery. For example, what is the lymph node status? In this case, we are using recurrence as a way to find some answers. And there are many other sub-classifications. I know there are 10 different sub-classifications that you can do. We have about 120 different clinical parameters that one can use. So, so forth, so on and so forth. Here, we focused, as I said, we focused on recurrence in triple negative breast cancer. And again, a little bit of context for people who are not, tuned into this kind of what. Triple negative is the hardest to treat tumor, because it doesn't have any of the receptors which are used generally for drugging. And it's known as a very, very aggressive kind of disease. And over and above that, the global average for triple negative breast cancer is just about anywhere between 8 to 12%. Somehow in India, this is about 28 to 35%, depending on which corner of India you are coming from. And doctors swear by it, that we see every third patient that we see turns out to be a triple negative patient. And this last 10 to 15 years, this has sort of gone on increasing. Nobody really understands what is going on, except that it's a hard to treat tumor. Nobody else across the world has reported such high numbers, except probably South Asia, India being one. Why is it so high? And the second thing that they say is the global average median age of detection in triple negative is like 52, 55. India, it is somewhere around the 35 to 42 bracket. Again, not really systematic studies. These are still hearsays, but I can see that people are putting this together, and the papers are now coming out trying to sort of just describe this difference that we see among patients. So, we had reasons why we focused on triple negative first, and obviously we had data or we had tissue which could be used for this. Now, with that, let me just give you a summary of what is really going on. So, after looking at that gene signature and doing, all types of regression analysis, what we are looking at is differential expression of genes between 14 and 76. 14 who had recurrence within 3 years, and 76 who are up to 5 years have been followed up and have been recurrence-free. Now, what Sherry's team was able to do was, and this is a little dated data, about 6 to 8 weeks back. Now we have a little better than this, but sort of will give you the idea of where we are going. So this 25 gene signature we could see was associated with disease-free survival in triple-negative breast cancer. We call this a protective signature. A Kaplan-Meier plot just tells you that these people have had no occurrence, and these are the occurrences that have been seen in the 14 patients. And you see these 25 genes have been able to discriminate between people who had recurrence versus who did not have recurrence very clearly. So what it is telling you that people with this protective signature are have very high survivals. So now how does one talk about this and say, let's first find out whether this is India specific or not. This goes back to the primary question that I was asked when I was sort of proposing this: how different or similar are we? So this is the slide that I used to answer that question. This is a Chinese data set, genome gene expression, and triple negative. Here recurrence is 49 and non-recurred is 311. This is Metabrick, a little older data set, but this is mostly microarray data, but very popularly used. And then, of course, TCGA is the gold mine, or the gold standard in this field, the TCGA data. And now we have used these 25 genes and asked that, how does it discriminate between recurred versus non-recurred, recurred versus non-recurred in different populations? And this is the Indian data. You clearly see that while it is being able to discriminate between this very clearly, it is not able to do it in other patients. So this is, again, a very small data set. I know the caveat. I don't want to sort of over emphasize on the importance, or because we still are about 14, and these are a lot more number of patients that we are talking about. But if you take all this together, it gives you an early sense of what is going on, and that is important. So we have tried to start focusing on this because it might take us another 5 years to find another 50 triple-negative breast cancer patients who have had 5-year follow-up. So till that time, we're hoping to sort of use this data and sort of build models and see whether we can sort of validate this more. I'll take a minute to explain this. This is called Cox proportional hazard analysis. What it does is, what doctors today, if you go to a clinic, doctors use tumor size, age of the patient, and the stage of the tumor, size, and this, to say whether it is going to be aggressive or not. And this is all triple negative. So if you have a triple negative disease, these are the 3 categories that doctors see and then decide how to treat, how aggressively to treat, how lightly to treat. Because these treatments, mind you, are really hard cytotoxic concoctions. They are always a combination of drugs. They're very, very strong drugs, and people have huge collateral damage. So the decision that is taken here about how hard to go on the drug, what is the frequency of the drug that is going to be given, is a very, very nuanced answer, diagnosis, which has a huge impact on the patient's lifestyle and quality of life. So now what we did is we took those categories and then said, now if doctors had information about this 25 protective genes along with this, what would perform better? So the Cox proportional hazard tells you that if you are here, it is the chance of misdiagnosis. It says that if you are in the group stage, depending on 2, 2A, 2B, 3A, 3B, these are stage of the tumors which are sort of done through histopathology. So it tells you that you would, the misdiagnosis in case of if you're here around the 0.5, it means 1 out of 2 persons could be misdiagnosed. If you're at 0.1, it is sort of 1 out of 10%. 1 out of 10 is misdiagnosed. And what is telling you that if you use the risk category, the chances of misdiagnosis is 1 out of 100. And when I'm talking about this, this is a very high significance also. The statistically things look rosy, and this is what we wanted to understand, whether this gene signature is giving you an added advantage over and above what a doctor would do in the absence of something like this. But I must also tell you that there is a good chance that low sample numbers are contributing to high significance. And I'm sure all of you here will be able to appreciate that, because you need numbers to come to great conclusions. You need great numbers. And 14 and 76 are not even beginners in that race. But look at the other side of the problem. The challenge that I have today is each of this patient costs about 3.5, 4 lakhs for the sequencing to come to it. The tissue has been stored for 5 years. All the clinical parameters have been taken about for 5 years till you come to this stage. The next 100 patients to add to this table might take 5 years, if funding doesn't come through, may or may not happen tomorrow. So what do you do in that scenario? So what we are doing is, we're taking this as a discovery cohort, and I don't have data on that, but I'm going to tell you. What we're going to do is, we are taking this gene signature and saying that we will go to formalin-fixed tissue. Formalin-fixed tissue and a gene signature of say 25, now it's a little less number, and also some genomic markers that have come from Nidhan's team. So we're putting together as a composite test, and we're saying that if we'll isolate DNA, RNA from FFPE tissue. FFPE tissue is a little bit easier to get, and more hospitals have that with more follow-up data. So hopefully we will be able to test this data, this discovery, validate it in a larger number of data sets from formalin-fixed tissue. Hopefully that will work out. And that is that. I'll just give a little glimpse onto where are we going with the, the multi-omics kind of fingerprinting that we are doing. Sort of taking, and people are talking about this quite a bit nowadays. A lot of papers and discussions and reviews are getting written where n is equal to 1. Each patient is a research problem, and how do you address that? So our 2 pence into that story is something like this. So what we are doing is obviously mutations, copy number amplifications, gene expressions. This is something that I did not talk about. We just started this actually, so there's not much to say also about this. Cell-free DNA, we collect blood from patients who have come through. So whoever agrees to share 5 to 10 ml of blood, we do that. We have just started methylation, and immune profiling is something that again I did not talk about. This is happening at mostly at IICB in Kolkata, and a little more than 100 clinical annotations that we have. So what we are interested in, and we have started putting together, is all this signatures into one, say something like a QR code, putting everything together into one fingerprint, and then saying that, can you use machine learning algorithms and deconvolute this data, and from this data now do not use any of the histopathological classifications that has been used before. Just go to molecular classifications. First make this fingerprints, then again deconvolute, break it up, and see if you can get back to cancer subtyping, molecular subtyping in this case, prognostic signatures which are predicted from here and along with this, and use that machine to forward predict what kind of gene signatures or what kind of fingerprints have prognostic value. And I'll come back to drug response in a minute in the next slide. I'll explain this, how we are looking at this. So the way we are looking at this is, we have started a lab which looks at this omics profile and looks at the patient profile. And for each of this, if you go to the pathway level from here, for each pathway there are at least 4 databases which have connected pathways to the respective drug that can be used for the pathway. So for a patient, if we have the profile, then we can say that this is the drug that can be used. A patient profile can be correlated with a drug profile. Now this tells you that if the pathway was this is the predicted drug. The predicted drug may not be the best drug that was actually administered. In many cases, we see that the drug administered is not actually the predicted drug based on this. But how does one test this? This is the lab that we have started about 2 years back. Now this is working pretty well. We have about 18 patients for whom we have developed the breast tumor organoids right from the patient tissue, both pairs, again normal and the adjacent normal and tumor. And now we are going back to the predicted drug and saying that what happens to this when we give the predicted drug. And especially when the outcome was not good, when recurrence happened, if we have tumor organoids for that, that is the way that we are trying to figure out, a much more difficult experiment here, but sort of validating the protocol and trying to see what we can get. So altogether, what we have been able to do, and this is just a peek into what else is going on. I've talked about methylation, cell-free immunotype. This is that cell type deconvolution from bulk RNA. Again, AI/ML for IHC. We have digital images. We've created all the immunohistochemistry that comes from the hospital are digital images now. So these digital images are being created. We're talking to actually a group in IIT Bombay for developing a large language modeling mediated application for germline context. We've had some discussions with colleagues here also on this, and eventually the idea is to sort of get to this n equal to 1 question and see if we can do better on this. So in the remaining time that I have, as I said, I'll talk very briefly about the lab questions that we play with in the lab. In contrast to this collaborative exercise that we have, this is, as I said, a much more intrinsic thing of the lab, and it has been developing over more than 15 years now. So what we're talking about is telomeres and how telomeres impact genome-wide mechanisms, and the case study here is going to be about, let me just go to the telomeres. Okay, so here is the end of the chromosome, telomeres. And what you'll be surprised of is the last 5 kb of our chromosome ends are nothing but this sequence repeated. Each is a hexamer, G3TTA. This hexamer is repeated for about 5 kb. The last 5 kb of your chromosome is nothing but this sequence. And the last 300 bases or 400 bases is again single strand, and that is the G single strand. Nobody really knows what this means, except in the 1980s and good into the 1990s, middle of 1990s, there's a lot of excitement in this area, mostly in chemists, who discovered that guanines, if you leave lying around in a test tube, form gels which cannot be shaken out. 1 millimolar solution of guanine, if kept for a few hours, you cannot shake it out. It forms a gel. The gel is because of this hydrogen bonded guanine situation, which is helped by sometimes sodium, potassium helps it. This guanine sort of get layered. But what happens is something like this. If you take each of this one set of guanines, let's say 1G here, 1G here, 1G here, and 1G here, make one plate. The second Gs can make a second plate, the third Gs can make the third plate, and very soon you have is all these NNNs are on the loops, and you have this very stable bookshelf structure. This structure is called guanine quadruplex. And this was a lot of excitement in chemists in 1980s, 90s, where they said that sequences can fold onto itself. And then now there are about 56 different shapes, sizes, flavors of this quadruplex that people study. And we don't exactly know what's really happening inside human beings, but except that there's enough information now to say that end of your chromosomes are nothing, might be arrays of these structures, because each of these 24, for this one, if this is one unit, each of these units will form one such bookshelf, and that will not DNA onto itself. So this array of quadruplexes make telomeres. This is a much more accepted thing in the last 5-7 years than it was before that. And what we started around in this thing was an exercise around early 2003, 2004, right after I joined. This was an exercise where we were trying to understand whether these sequences are restricted to telomeres or do they occur elsewhere also. And we were really surprised by seeing that not only do they occur outside, they are also occurring not in a non-random fashion, some here, some there, thrown about, but they are focused on promoters. These are all tied onto the transcription start site, and each row here, if you can imagine, is a chromosome. All the transcription start sites on that chromosome are tagged here. So it means that if you were counting G quadruplexes, you would be going up a hill as you reach a promoter or the transcription start site, and as you cross the transcription start site, you will be counting coming downhill. So this is more or less a representation of that. And more importantly, these are conserved across species, and this is human, chimpanzee, mouse, rat. And we have about 1,200 more microbes that we studied around that same time, and we found that to be true in microbes also. But we were really punctured. All our enthusiasm was gone when the paper came back with this. Somebody called it, these are accidents of genome, forget it. There are so many evolutionary accidents that have happened, and this might be one of those. There's nothing called DNA secondary structure, or at least quadruplexes that exist in genomes, because there's no function associated with it. Well, what it egged us to do was go into function. And that was interesting because we started doing now experiments about trying to understand how intact G quadruplex was necessary for gene regulation, and later on also into epigenetics. So we showed that in the first transcription factor we were sort of egged by this kind of statements to go on and move, and that helped us discover the first transcription factor which binds to the structure, because we have shown that if you use small molecules, you can exclude this binding and it impacts gene regulation. Or if you can genetically disrupt the quadruplex, which is very much possible also, again you don't see transcription factor binding and it goes off, and that led to a number of papers. But I also don't want to leave you with the note that this is all us doing this work. No, some of this is yes, and this is where we started 2003-04, and about 100 papers, about 5 to 6 groups around this time, and now I see about at least 50, 60, 55, 60 groups working on this, and the papers have also sort of. So all of these are G quadruplexes and their function in terms of epigenetics, initially gene regulation, epigenetics, now into recombination, DNA break repair, and many different things. But we went on and tried to understand, telomeres. All of us know we lose telomeres as we age, and there's also a lot of public material on telomeres. Telomeres keep you happy. Telomeres keep you young. If you can take care of your telomeres, they take care of you. Many such and very noted authors also. If you can read something, this is Elizabeth Blackburn and another colleague of her who wrote this. It makes you live younger, healthier, longer. Somebody called them telomere time bombs, because if it shortens too early, it's like a death sentence, and so forth, so on and so forth. So keeping all this in mind, one thing really troubled us. What is the impact of telomeres on gene regulation? If you come with this focused question and go into literature, few years back you would have discovered just this: telomeres can loop to the peripheral regions, which is known as the subtelomeres, which is about 10 MB from telomeres, a tiny bit outside telomeres. It can loop and it sort of heterochromatinizes, because the telomeres are deeply heterochromatinized. So the heterochromatin spreads and these genes are repressed. That's it. So if you've talked about so much of physiology which is associated with telomeres and called them time bombs, why is there nothing known about telomeres impacting anything in the rest of the genome? So this was telomere positioning effect. This was both Jerry Shea and Woodring Wright are famous for proposing this and showing this. Telomere positioning is this. We started asking this: what happens to the rest of the genome? And let me just take you through this. Like the ends of shoelaces, telomeres have to be protected. Shoelaces are protected by plastic clips. These are protein clips, complex of proteins which puts the clip, because without that it starts fraying. And this is also known because it shelters the end. This is known as sheltering, the sheltering complex. To answer that question, that what are telomeres doing in the rest of the genome, we needed somebody to test, somebody to play. Who is the central character of the plot? So we focused on this protein called telomere repeat factor 2, TRF2. Very well known in the telomere field, very famous, because this was the factor which was taken out and shown that telomeres fuse. Why? Because this complex ensures that telomeres are not detected as double-strand breaks. Because if a double-strand break is detected, the whole policing machinery which comes into play and seals. And this is exactly what happens. You take out TRF2, 2 chromosomes are sealed, because it is discovered as a double-strand break. Now we took this TRF2, and the first question is asked is TRF2 only here, or do you see it elsewhere? So positioning of TRF2. This led to a very interesting paper where we showed that TRF2 exists at dimers. We showed that not only does TRF2 bind to G quadruplex, but there are more than 20,000, sometimes 40,000 sites also, which depends on cell lines. We found that these are outside telomeres. You have so much of TRF2 sitting outside telomeres. And somebody picked this up, and we are very happy to see a couple of years later, people say that, from the wings to the center stage of chromosomes, TRF2 has moved from the wings. For 40 years, it was known to be at the wings. Now it has sort of moved. So they did a great service to us by, publishing this paper from Japan. because it sort of brought this whole idea into the field that TRF2 can have a second home. But what happens to TRF2 dependent transcription in long versus short telomeres? That was the actual question we wanted to ask, because telomeres shorten with age. Now, if telomeres shorten with age, can there be a molecular impact which is distant from telomeres? So in trying to answer this question, Ananda and Shalu collaborated on this work, and I'm summarizing a lot of work here. So what we did is we make cellular models, just engineered cells. These are very artificially engineered cells, but different cell types, where we decreased TRF2, decreased telomeres, increased telomeres at our will. We got really good at it after a while in increasing and decreasing, because everybody started playing with the ways of doing this. But what this helped us do is, when we have already shown that TRF2 occupies many regions, including promoters, then what happens is when it shortens, when telomere shortens, this TRF2 can increases on the promoters and vice versa. Obviously, this is possible if simple biophysics tells you that this is possible if TRF2 is not copious. It is limited amount of TRF2 is present inside cells, which is true. Only limited amount of TRF2 is present. It is not there. It is not a sea of TRF2 out there. So therefore, this partitioning works. And when this partitioning works, we are able to also show that TRF2 is not only by itself, it brings in histone modifiers, and it helps in repressing chromatin, mostly repressing. About 20% of genes were actually activated, and we had a different way of figuring out what happens. It's about TRF2 being, post-translational modification of TRF2 being different leads to activation or repression. Mostly repression, so this schematic catches that. And so this sort of brought this idea into the field that TRF2 redistribution is telomere length dependent, and therefore it can impact gene expression across the genome. And genes which are 60, 80, we showed about 82 MB away, few genes which are about 60 to 80 MB away. That's what it is. And also epigenetic, because this brings in some of this epigenetic modifiers. So this is what we capture in a nutshell. This is what Woodring and Wright had proposed, the telomere looping and telomere positioning effect. If telomere shorten, this is repression is taken off and these genes get repressed. So we covered this. We said, that's not the full story. Telomeres expand their sphere of influence, and we said that the molecular impact of telomeres is like in some ways like an earthquake. It is felt way, way away from where actually the event is happening. So some of the molecularities we explained, but not all, some of it. So then we started people started asking, this is all engineered cells, it's great, it's engineered cells and it's great to show that something works, this model probably works. But where does it work? What is the physiology that is connected with this work? So these are the questions that started from there. This is the work that I'm going to talk about, telomeric autoregulation. But few others, this is now in E-Life, you can telomere-sensitive interleukin signaling. This contributes to immune suppression in the tumor microenvironment. It's a very interesting story here. This is the story in the almost done, but what we see is telomeres also contribute to how neural stem cells differentiate. And that's another interesting way of looking at what happens to neural stem cells as you age. Why do they differentiate more with aging? And stuff like that. So let me come back to this story quickly. But I would have to spend 30 seconds on telling you what is telomerase. Telomerase is a protein, only protein, which can make telomeres. And that is why Nobel Prize was given just for the idea that they first proposed and then they discovered. They first proposed that a special telomerase would be required for making the telomere ends, and then showed that that special telomerase is this. They used a Thermophila, a different organism for this. But yes, so what it does is telomerase has an RNA component, and that is the template. This is called HTR or TERC sometimes, RNA template TERC. So this TERC actually forms this is the end of the telomere. This is the template, and telomerase comes and puts this using this template, it synthesizes the telomere, and then again hops on to the next bit. And that's how telomeres get synthesized, by a single protein called telomerase. Now we started noticing that at the transcription start site, and this is of the telomerase promoter, transcription start site of telomerase had quite a few quadruplexes. And this quadruplexes attracted our attention. And we said that if this is true, is it possible that TRF2, by this time we knew that TRF2 binds avidly to quadruplexes across the genome. So obviously this question came to our mind, if TRF2 binds, yes, it does bind. And this is published in this paper, so I'm just summarizing this data. This is in Cell Reports 2021. So it not only brings, it brings the canonical repressor complex. EZH2 is the methylase, methyl transfer here, and then rest, and this is the full PRC2 complex. TRF2 brings the PRC2 complex. The PRC2 complex brings in the, the TRF2 G4 association via PRC2 complex, keeps the telomerase promoter repressed. And we showed it till this much. Then the next part of the work started with Antara's work, where she said that, when she asked us if telomerase is, this is what telomerase does, it expands, why is the telomerase expression going up and down? You see really going up and down. It sort of maintains something. As soon as you reach, stem cells have very high telomerase. Normal somatic cells have almost no telomerase. It's sharply repressed. But before, in tumorigenesis, just before crisis, it again is overexpressed, and it is really overexpressed in cancer cells. So there is this ping pong going on with telomerase, and it is almost deciding the fate of cells. So there was a question there which Antara got interested in: is telomere length having anything to do with telomerase expression? Knowing that this is the only protein which can make the telomere length, it looked like an interesting question. So she made her models which asked this question, that if I increase or decrease telomeres, what happens to the telomerase expression, right? And she recently defended her thesis, so this is a picture from there. So we started with making this short telomere, long telomere models. One is obviously you overexpressed hTERT, but then we wanted hTERT independent, because we were part of the study is also about hTERT regulation. So this is exogenously overexpressed hTERT, and we will look at the endogenous TERT expression in a minute. And these are long and short telomere. Again, you can take away the RNA component. You can silence the RNA component, and that will again lead to lack of telomerase, which will give you short telomeres. And you can do a very interesting, more modern way of doing things. You can make short telomeres by just clipping using sgRNA. You can clip off some of the telomeres, and you get short telomeres. So all said and done, these are two different cell models, and you see we achieved this short telomere for ST. I keep on using this ST and LT reference. ST is short telomere, LT is long telomeres, and this is a facts showing that this is the telomere signal has increased in long telomere cells. This is blue LT cells. Again in here also. So these two models we have here, and this is just to convince you that telomeres are really increased or decreased, and this can be caught very clearly with facts. Now, what happens to endogenous TERT expression when telomeres change? Where are we? We are in the early promoter. This is 0 to 200, 300 to 450, all the way to about minus 750, TERT, upstream TERT promoter. GAPDH is the control. Again, ST is short, LT is long. The expectation is, if telomeres are long, TRF2 is stuck to the telomeres and lost from the promoter, or vice versa. If you shorten telomeres, TRF2 comes back on the promoter of TERT, and it is a repressor. So mind you, which means if telomeres shorten, TERT will decrease. If telomeres elongate, TRF2 is pulled away, and therefore the repression is lost and TERT increases. So what we see is promoter activity here. The promoter, this is occupancy of TRF2. Exactly what we, as I said, in short telomeres versus long telomeres. Short telomeres have much more occupancy around the early promoter, between 300 to 450. You see higher amount of TRF2, and as we already had shown that TRF2 just doesn't come by itself. It brings in the whole bulldozer, which is the repressor bulldozer, and therefore you expect that telomerase will shut down. This is hTERT mRNA. Exon 7, 8, and 15, 16 are being studied here, and we see that telomerase, as in long telomere cells, telomerase has increased so the regulation is kind of working here. This is a little bit more intricate temporal model where you don't do this shortening business and engineering. You use a DOX-inducible TERT, and what you do is you start DOX, which induces TERT, and then after a while you stop DOX. So up to 10 days you give DOX, telomeres increase, and day 10 you stop DOX, and the idea is telomeres should fall because telomerase expression is going down. So this is the facts for this cell, day 0, 6, 10, 16, 24. This is the timeline. Mind you, at 10 we are stopping doxycycline. So it's supposed to increase till here and then decrease. Day 0, day 6, not much difference. But day 10, day 16, we have stopped at day 10, but just increased a little by day 16. By 24, it has gone down to almost its natural levels. So telomeres can be shortened, and Now we want to follow these cells and see if as telomeres increase and decrease, what happened to the actual telomerase expression. This is telomere length. So our model had predicted this long shot should also impact telomere. So this is TRF2 occupancy on those cells. And again, a little bit of, I'll put circles on here. Yeah. So day 10, right here, day 16 here, and day 24 here. What is happening between 16 to, as you see, it increases till day 16. So as it increases, you want TRF2 is going away. So it should fall. It falls. By day 24, it should come back. It comes back. We're a little surprised by actually this. We're not expecting so clean. So this was repeated in, this is HT1080. This was repeated in 3 different cell lines, HCT, MRC5, and then we took HEK293. Very, very laborious experiments, but always we see the same. Believe me, always we see the same thing. It does work like that. And it doesn't, not only is the TRF2 occupancy changing, the TERT, here we are showing you the 3' UTR, just to keep it away from the, this is exogenously expressed TERT. So you want to look at the endogenous TERT. This is the endogenous TERT. You see it increases as TRF2 falls, increases. As TRF2 comes back on the promoter, the repressor function kicks in, again it decreases. And it is reproducible across cell lines. So the summary of this is, again, this is just a, sort of giving you the idea that, as TRF2 occupancy at the TERT promoter alters with telomere length, as telomere length changes, this alters. As it alters, if promoters are short, there's more TRF2, it brings in more of the repressor complex, and therefore, the epigenetic marks 27MA3, we showed this, all this 27MA3 increases, and TRF2 falls down. So the basic takeaways from this. There's a molecular link between telomeres and telomerase, only gene which makes telomeres. So this is sort of intuitive. Well, there should be a feedback loop, but I don't think anybody had looked into this in so much detail. And telomeres elongate, more TERT is produced. It is required, if telomeres elongate, it requires more telomerase to keep it, maintain it. After every cycle, the exact length of telomeres has to be remade. So it requires more amount of telomerase, and that's what happens. And in aging cells, telomere shortening represses TERT, as is represses, and epigenetically targeting. So what we have also done is, we have epigenetically targeted the TERT promoter, and by doing that, this is the important part, one can delay cellular senescence, because, senescence kicks in at some point, and if you can artificially work on the TERT promoter, you can actually delay senescence of cells. By how do you go to the TERT promoter? I was talking to Dilip today morning about this, today afternoon. So what we can do is use a guide RNA, but on the dCas9, you take a Cas9 which is dead, which cannot cleave. You fuse a transcription factor to that, and now use guide RNA to take it to the TERT promoter. Now you can play with the TERT promoter as you like. So we did exactly that, and one can start doing these things. I think I don't know whether within time or not, but hopefully I've been able to talk about some of the excitements that we have. Antara and Saujanya were the main players here, and really a lot of collaborators without whom I cannot stand here. Arindam, we work on a project on neuro, which I did not talk about. Arindam is a part of that work, and Nidhan work I've discussed. Many of the others here are contributing in different ways, and thank you for your patience.
Thank you, Dr. Chowdhury, for this insightful oration. May I please request our director, Dr. Sagar Singh Gupta, to kindly felicitate Dr. Chowdhury.
Usually oration talks
Don't have questions, yes.
but if you are wanting,
I am happy to. I am happy.
You have interaction with the students, so they will be able to ask you as many questions.
Whatever is the protocol and whatever, I'm happy to. Either way. Wow, thank you so much. He knows I like this. Yeah, this just shows that we know each other for too long. Oh, thanks so much, Sagar. It's a pleasure. Thank you very much.